So last time we ended with transcription rather, and now we're going into the post-transcriptional modifications, which to be noted largely happen in eukaryotes. So just a short recap. Remember that in eukaryotes we have three polymerases. We have RNA polymerase 1, which transcribes our RNA or ribosomal RNA. RNA polymerase 2, which transcribes our messenger RNA, and RNA polymerase 3, which transcribes our tRNA or transfer RNA. So for both tRNA and rRNA, our RNA goes through trimming, meaning if you have a transcript here, the ends can be trimmed. You can also add terminal sequences. So that's usually if you want to protect the ends of your RNA from nucleases or from exonucleases. And in some instances, base modification happens, such as if you methylate a part of the base or etc. We're going to focus on mRNA post-transcriptional modification. So here, shown is the DNA strand. And if you will see, there are exons and there are introns. So exons are usually the coding regions, meaning they are the parts of the gene that get into the final mRNA transcript. So they're the ones that encode the protein. In between the exons are the introns, and these are the parts that usually function as regulatory segments, so which basically means that they don't encode for a protein, but they are important in regulating transcription and other processes. So if you will find in eukaryotic mRNA, so here it's the first transcript, our genes are discontinuous. So exon 1, 2, 3, and 4 encode for a single gene. As such, eukaryotic mRNAs are usually called monocistronic. Mono meaning single and cistron meaning gene. So with eukaryotics, you have a single mRNA to encode one protein. Whereas in prokaryotes, a single mRNA can encode several proteins because they are polycistronic in nature. So here you have your mRNA transcript and we're going to process it. So one of the ways with which we process it is by splicing. So what splicing does is that it removes these introns rather, so that you can have a continuous gene in the final mRNA transcript. So these, the first transcript or the primary transcript is also called hnRNA or heterologous nuclear RNA. It's also called the pre-mRNA or the precursor mRNA. So after you've removed the introns, so you now have all the exons remaining, we also add in a poly-A tail. So it's a tail that's largely composed of all adenines and a 7-mg cap or a 5' cap. Now what the capping and the tailing does is that it protects the mRNA from exonucleases. So we make sure that they reach the ribosomes intact. So here is the 5' cap. So if you will find, this is your guanine, this is the sugar group, and this is its phosphate group. This here is the 5' base of our original mRNA. It's 5' end, and here is the capping. So there are two phosphate groups, the sugar group and then your guanine. So altogether they form a triphosphate linkage. which is unusual for exonucleases because exonucleases can only act on a monophosphate or a phosphodiester linkage. Whereas the one shown here is a triphosphate linkage. That's why it makes the mRNA resistant to cleavage. So, how does splicing happen? So here is a video that shows splicing.
Eukaryotic genes typically contain introns, which have to be removed after transcription. Before the RNA transcript leaves the nucleus, the cell splices out the intron sequences. A few short nucleotide sequences provide the cell with cues of what to remove. The elaborate molecular machine that carries out this task is called the spliceosome. A branch point binding protein, BBP, and a helper protein, U2AF, recognize the branch point site within the intron.
Okay, just to note, these BBPs, U2AFs and U1s, you don't have to memorize them. The point of this video is just for you to appreciate the process. We're not going to memorize all the components in here, but it is important for you to note what a spliceosome is.
And an RNA and protein complex, called a SNRP, recognizes the 5' splice site by forming base pairs with it.
So the SNRP or the snRNP means small nuclear ribonucleoprotein. So that is a combination of your snRNA and a protein group. So if you will remember from a previous lecture, snRNA functions as a ribozyme, and it is important in post-transcriptional modification. Specifically, it is important in splicing. So whenever you encounter a protein that has a U on its first in its first letter, so anything that has a name of U here in splicing actually refers to a SNRP.
Next, another SNRP base pairs with the branch site, displacing the bound proteins. Additional SNRPs now come into play, and several RNA rearrangements occur to break apart the U4-U6 base pairs and allow the U6 SNRP to displace U1 at the 5' splice junction. Now in position, a conserved adenine nucleotide in the intron attacks the 5' splice site, cutting the sugar phosphate backbone of the RNA. The end of the intron covalently bonds to the adenine nucleotide, forming a lariat structure.
So a lariat structure means a loop.
The spliceosome rearranges to bring together the exons, allowing the 3' hydroxyl group of the first exon to react with the 5' end of the other. After the two exons are joined into a continuous sequence, the lariat is released and degraded.
So now you have a continuous mRNA after splicing. So I'm just going to write down the words used during the video. That would be your spliceosome, meaning all the protein machinery needed for splicing to occur. You have your SNRPs or snRNPs, which mean small nuclear ribonucleoprotein. I'm sorry for my very bad handwriting, which is a combination of your snRNA plus a protein component. Okay? Now, there is a phenomenon called alternative splicing, meaning if you have a gene here and it has 6 exons, so you have 1, 2, 3, 4, 5, and 6, it is not necessary for all the exons to go into the final mRNA transcript. It is possible that some exons will be spliced out. So for example, in this first protein, you only have exons 3, 4, 5, and 6, whereas in this protein you have exons 1, 2, 5, and 6. And because these proteins are encoded by the same gene, we call them isoforms or protein isoforms. Now, it was said that these proteins will have the same function except that they are recognized by different organs. So, for example, IL-1F7A is recognized by the heart, let's say, whereas IL-1F7B is recognized by the brain. So, which means that even if this isoform, gets into the brain will not respond to it because it only recognizes this isoform in the brain. So, which means that some proteins have specific actions towards certain organs only. So, alternative splicing is also one of the reasons why, even if you have a single mRNA transcript, you can get, in this example, 5 proteins. So, this explains why when scientists tried to quantitate how much proteins you get, they are more than the number of genes in a person. So that's because of alternative splicing. Next, let's go into transcription regulation. So there are several ways to regulate transcription, and some of them involve enhancers and silencers. Enhancers and silencers are DNA sequences. Okay, they're not proteins, they're DNA sequences, and they're usually found upstream the transcription unit. So upstream your promoter. So in this example, you have your enhancer here, which is recognized by a protein. This protein has an adapter protein or a mediator protein, which recruits all the transcription factors needed for transcription to occur. Now, because of the presence of this mediator, it makes the transcription factors bind tighter to the promoter, hence enhancing transcription. On the other hand, if you have a silencer, so the silencer is recognized by these proteins here, and when these proteins bind, due to steric hindrance, they prevent the binding of the transcription factors. If your transcription factors are not able to bind to the protein, then transcription will not happen. Next are response elements. So response elements are specific for certain conditions. Like, let's say if you have a very high temperature, you can have response elements that are specific to temperature. So that means that these regions, when they are activated, they will enable the transcription of proteins that are necessary for temperature control or to address that temperature. So this is kind of like your playbook. So in the body, you have a playbook such that if you're placed in a certain situation, you will know how to respond to it. So that's what the response elements are for. Next, you have non-coding RNAs. So there are two examples of non-coding RNAs. We have microRNA and small interfering RNA, which, if you will remember, participate in a process called RNA interference, which basically what they do is they bind to the target mRNA and label that for degradation so that your mRNA does not go into the ribosome and it will not get translated. So there, inhibits translation. And that will be the end for post-transcription modifications.